Right, good afternoon, ladies and gentlemen. We will be starting this in just a minute. We're just going to wait to see if we have a few more people attending, and then we will get straight onto it. Thank you. Okay, for those of you that have joined us, this is a talk about ultra-fast point-of-care MDX development with new sensitive lamp master mixes. So what I'm going to do, I'm going to give you an introduction, first of all to the company, just very, very briefly. Then I'm going to give you a little brief introduction about point of care and the advances in point of care recently. And then I'll hand over to my colleague Elda, and she will actually carry on. So what we'll do first of all is, unfortunately, the moderator has been delayed, so she won't be turning up. My name, I'm Dr. Steve Hawkins, Product Marketing Manager, and as I say, I'm going to introduce it, and then I'm going to hand over to my colleague Elda. Hello, Elda.
Hi, Steve.
And she's going to then continue, and she's going to talk to you about the lamp technology. So for the agenda itself, there'll be just a brief introduction to Meridian Bioscience, then an introduction to point of care, and then go through all of the lamp information, assay design, time to results, sensitivity, specificity, etc. And then we'll have a conclusion, and we'll also have questions. Just before we start, what I would like to do is to first of all point out that if you have any questions during this, you can actually write the questions in the section on the side. And whilst you're thinking about this, we're going to have a couple of polling questions. So the first polling question I will launch now. And what we'll do is we'll just have it on very, very briefly. What we will then do is we will just continue. And all I really want to do is I just want to find out how you actually found this webinar, whether it's by email, LinkedIn, Facebook, Twitter, or a friend. So just going to take just a few more seconds just to leave it on, and we shall close it there. And just to let you know, most of you, oops, apologies, most of you actually came on by email. So that's great to see that our emails are getting to you, and yeah, you have actually joined us. So welcome to the webinar. So we'll continue. First of all, Meridian Bioscience. Meridian Bioscience itself is approximately 45 years old. We have over 800 employees now, and that's in over 13 locations worldwide, all the way from the US in Cincinnati, where our headquarters is, all the way through to Australia. So all different places. We're in lots of different markets, over 70 global markets worldwide. The company itself is split into two, a diagnostic side and a life science side. So the diagnostic side focuses on gastrointestinal, neonatal, pediatric and respiratory conditions. And then we have the life science side, which is the side that we're involved in, that has immunological and molecular assays. And it's the molecular assays we'll be talking about today. And last but not least, just to mention that we are in several hundred different registered assays. So everything from COVID assays, over 70 COVID assays, all the way through to cancer care, to blood banking, all the way through to environmental testing, even vet markets. We have many assays in vet markets. Now what I'm going to do, I'm going to introduce you to the molecular point of care. So traditionally, molecular in vitro diagnostic tests have always been centralized, and there are several reasons for this. But one of the issues with this is, first of all, it does actually delay the results. You need to send your information, your materials off to the centralized lab. It needs testing, it needs to come back again. This, of course, also produces a high cost for the tests. It also can be less accurate as well. If material is being shipped backwards and forwards, there's time involved, it could degrade. And last but not least, you're going to get later diagnosis, and this could actually improve the mortality rate. So if you'll just bear with me just a second. We don't seem to be changing the slides over. So let me see if I can improve this. So I'm not sure whether the slides aren't changing. And we seem to have an issue here. So see if I can just restart it, and hopefully you should be able to see the slides changing. You can see the slides changing now.
Yeah.
Very, very good. Sorry, I apologize about that. So we'll go back to talking about the traditional molecular in vitro. As I say, it's centralized, and the problem is that you delay the results. This potentially increases the mortality rate. It also has hindered the growth of the market. In recent years, however, there have been changes, and point of care testing has really started to become properly commercialized, particularly in settings such as areas where timing is critical. This is like emergency triage, or it might be, as we've seen with the COVID, with infectious diseases where you need results as quickly as possible. Also with cardiology monitoring. So this is looking at somebody having a heart attack. You need to know if they've had the heart attack really quickly so you can administer the right medicines. And also with things like oncology testing, the earlier you can actually tell not just whether they've got cancer, but what type of cancer they've got, the better the prognosis. So this has really driven this technology. We also have where the lab facilities are distant. So you might be in a physician's office, or you might be in an urgent care facility where you need the results very quickly, but there's no labs available. Equally, you can go even further where the laboratory facilities are completely non-existent. And this may be in things like a military setting, or in developing countries, or simply just out in the community. Now, this has, and we've actually shown that with good testing performance, the point of care approach helps deliver appropriate and prompt treatment and helps protect the patients, improving clinical outcomes and thus significantly improving the reduction in things like expenditure and, of course, saving lives. So, in spite of such drivers, there have also been concerns about the reliability and the benefits which have impeded the point of care from achieving its full potential. And these include things such as not having universally accepted standards. And this is actually improving. And again, with things like the COVID, we've actually had to come up with certain standards, and this has been shown with the FDA and some other approval systems. So it's being worked on and it is improving. Testing is often performed by personnel without training. So again, COVID, I'll come back to this because it's a wonderful example where we had volunteers actually going and doing the testing. So that's been a point of concern. And thirdly, even more of a point of concern is the self-testing. So this is the patients themselves doing the testing. And again, there's little control over this. So there needs to be a balance there of working out just how they can do it and how easy it's going to be so that they can make sure that they do it correctly. Last but not least here, there's also certain clinicians favour traditional centralised testing. So they'd rather have it sent away and tested properly than them testing it themselves and getting the results. So these have been concerns. Having said that, despite of all of these and potentially the difficulties gauging the cost effectiveness, I mean, if you're comparing a point of care which may look expensive with sending it away to a laboratory, if you send it away to the laboratory, there's the cost of actually getting the material, then sending it off, and then the person in the lab actually doing the testing, then sending it back again. So there are hidden costs within both of those, and it's very difficult to gauge how cost effective it is. But despite that, there have been a lot of tests and a lot of research done that actually shows that there's measurable improvement in the workflow efficiencies and patient care. And numerous studies over several years have actually shown that it really does work. And of course, the pandemic with the asymptomatic spread of the COVID has really highlighted this. So some of the improvements in the point of care itself, there have been recent advances in the development of, first of all, genetic sequencing. And I'm not just talking about sequencing the human genome. We've had the human genome sequenced for 20 years now. But what we have done in the past few years is we've sequenced more and more genomes from different sections, and we've been starting to do comparisons. So we can segment it, put people in segments and actually show whether they're going to respond to treatments, whether they're going to be better on a certain regime than another regime. So this genetic sequencing has actually really helped. Secondly, there's microfluidics, being able to move small amounts of volumes around very, very easily. And thirdly, of course, portability. You need portability if you're going to have a point of care instrument. And these have led to a rapid expansion of this market, particularly, first of all, in oncology. And oncology is very, very important because we have an ever-aging world population. And as the population ages, so you're going to see more chance of people developing cancers. And what we need to do is, first of all, we need to be able to see what those cancers are, being able to make sure we administer the right drugs, and being able to catch them just at very early stages so that we know the prognosis, if it's caught at an early stage, is going to be better. Secondly, with infectious diseases, and this is even before COVID came along, we saw a big expansion in this molecular point of care with things like streptococcus, with influenza, with RSV, and with other diseases as well. So if we look at the types of point of care assays, first of all, there's genetic sequencing based. Although the analysis and the interpretation, they still tend to be an issue. There's a lot of information you get from genetic sequencing. So a lot of the time, these still tend to be based within laboratories, and the information is sent back. We have hybridization based. These are very inexpensive, but the problem here is the accuracy, and particularly the reproducibility as well, is often very hard to achieve. Thirdly, there's microarray based. These have the capacity of doing very, very high parallel, high throughput monitoring. But here again, you have a problem that you need highly skilled personnel, you have a high cost, and you also have a prolonged procedure. And even then, when you get the results, you need to interpret those results. So this leaves nucleic acid-based assays such as quantitative PCR, so qPCR, and isothermal amplification, in particular, things such as loop-mediated isothermal amplification or LAMP. So if we concentrate on those two, the qPCR has always been seen as the gold standard, and it's been the main focus for the molecular point of care. And this is due to its sensitivity, its specificity, and its simple quantitation methods. However, again, since COVID first started, what we have seen is we have seen LAMP really take off. It's because it uses a single temperature, it's very easy to use, you can have simple detection methods, and not only is it starting to catch up, but in some places actually overtaking qPCR as the point of care method. So having introduced you to that, what I'm now going to do, I'm going to hand you over to Elder, and she's going to talk about the technology itself. So over to you, Elder.
Thank you very much, Steve. So, hello everyone. So, as you mentioned previously, Steve, so LAMP technology over the last decades has played an important role in the molecular diagnostic. So LAMP has the advantages because it's rapid, sensitive, specific, isothermal, and has less instrumental constraint. But the other side has the advantage, the disadvantage is that the assay design, it can be a bit more challenging to design. So LAMP compared to the PCR use 4 main primers and the addition of 2 loop primers. So it can vary from 4 to 6 primers. And the addition of the loop primers, so you will need a LAMP, you use it to have it like more faster reaction. So the primers, so the distance and the number of the primers are recognized from 6 to 8 region of the unique sequence of the DNA which you need to amplify. So there are software which assist in the primer design, but the output of this software is like the 4 main primers, and the loop primers you still need a bit of optimization. So you need to design different sets of loop primers and to make an optimization for primers to don't generate primers with slow amplification, poor sensitivity, or not specific amplification. So, however, so LAMP has a variety of detection. So you can use a fluorescence by intercalated dye or fluorophore labeled primers. So you can use a colorimetric assay just by naked eye. You can distinguish positive and negative amplification, chemiluminescence, or turbidimetric method. So as you can see, so LAMP has a variety of detection, which make LAMP technology more ideal. It makes it ideal for the point of care. So LAMP has a factorial amplification and makes LAMP itself faster than qPCR because it's an isothermal, so there is no cycling of the temperature, so LAMP itself is really faster depending on the template as well. So LAMP can be multiplex as qPCR, but in LAMP it's normally it's used like to 2 or 3 targets. So increasing the number of targets, you will increase the number of primers, so that can bring in primers dimer or interaction, and you will have the issue of not distinguishing the amplification with a specific or not specific amplification. So, sorry about that. So at Meridian, so we developed great expertise in designing LAMP and RT-LAMP master mixes. So the master mixes, they can be air dry or lyophilized. And so all the master mixes, which are they can air dry and lyophilize with primer as well, depending on the assay. So the cake, it can be stored at room temperature for at least 24 months. And this changed completely the way of shipment. So you can ship the air dry or lyophilized master mixes just in the room temperature. You don't need more the cold temperature, and the cost of the shipment completely change. So having the master mixes air dryable and lyophilized, so it's it opened the room to air dry and lyophilized master mixes in cartridges, which make this ideal for point of care. So from the few slides from now and the other slides, so I will show some the master mixes what we develop at Meridian. So here we are showing that the master mix, air dryable master mix for the detection of RNA and DNA LAMP mix. So the master mix, the air dryable, was air dry with specific assay for Epstein virus or for RNA bacteriophage virus. So MDX 180, which is an air dryable, you can see in blue, showing an amplification of 3 technical replicates, and it's showing like the faster amplification compared to one of the best supplier in the market, which is the NEB. So there is like a distance, like the difference from 10 TTRs from the amplification of DNA or RNA specific. So this was like on the with 1,000 copies of the template. So this is the master mix specific for RNA DNA, and it's an air dryable master mix. Regarding to lyo mixes, so the lyophilized mixes is that we developed the lyo ready LAMP mix, which is for DNA detection. So the Lyo Ready was lyophilized with specific primers for Trichomonas vaginalis in this case. And so the master mix, the MDX097 shown in blue, was compared to the Optigene and NEB, to the two suppliers in the market. And you can see that with the master mix, the Lyo Ready shows a faster TTRs compared to the Optigene and NEB. This regarding to 1,000 copies template, 100 copy, 10 copy template. And for 10 copy template, so the amplification is so fast, it's like less than 10 minutes, and has a greater reproducibility compared to the Optigene and NEB. So the Lyo Ready lamp mix was developed for DNA. Regarding the RNA, the slides are not changing. Regarding the RNA, so we developed the mixes which are Lyo Ready RT lamp mix. So the Lyo Ready RT lamp mix was compared to the NEB warm start, and it shows faster TTR to the NEB. In addition, the Lyo Ready RT lamp shows that it can tolerate inhibitor tolerance, which can come from universal transport media, sputum, or saliva. And saliva can go up to 25% of the human saliva. So the next step, what we developed in Meridian, was to make it the master mixes, the Lyo and air dry master mixes, more inhibitor tolerant, to make it more greater inhibitor tolerant. So as I'm showing here, it's a Lyo Ready direct DNA lamp saliva, which means that the master mix is lyophilized and is resuspended with saliva directly with a specimen. And here we are showing like an example of 3 different assays from cytomegalovirus, adenovirus, or herpes virus. So the master mix, the Lyo Ready master mix shown in blue, has an early TTR compared to the NEB, the supplier in the market, and they show like 30% of inhibitor tolerant to the saliva, universal transport media, or viral transport media. So the master mix was for the DNA. Regarding the RNA, so we developed a master mix which is called RNA DNA lamp saliva, and the master mix was lyophilized with a specific assay for tuberculosis, and was detected in, you can see like with the Lyo mixes, you can resuspend it with saliva, with different concentration of saliva, and the master mix can tolerate up to 60% of the saliva. So both the mixes, the air dryable and lyophilized mixes, so they can be like, they have advantages because they can be used in wet or in dry form. So in dry form, they can be used dry form itself, but plus they can be dried with probes or with the primers with different assays. And having the dry mixes, and these are like for point of care, are ideal for point of care because you can increase the volume of the sample. Like in this case, like we managed to increase the volume up to 60% of the specimen, which this brings in the increase the fold of the detection, the sensitivity of the detection. And plus, if you use the direct sample, so you don't need more RNA and DNA extraction. So both the master mixes, air dryable and lyophilized mixes, so they can be stored at room temperature, and they can be stored at least for 24 months. And here, like for instance, like you can see like the different from lyophilized and wet mixes, so the performance doesn't change. And the same for air dryable master mixes. So Steve, so yeah, just to summarize, so here at Meridian, so we develop a high-con glycerol-free BST, which can be used for isothermal amplification. So after that, we focus on the buffer, so to make it buffer glycerol-free, so which are it can be ideal for air dry and lyophilized. And having the master mixes air dry and lyophilized form, so we focus it in making this master mix it more inhibitor tolerant. So yeah,
right.
Thank you.
Thank you very much. That was great. Very interesting. So what I'm going to do is I'm going to open it up to the audience to ask questions. So I will say just before we open it up to the questions, first of all, we do a number of different polymerases and buffers that actually go around the system, as well as the mixes. So if you are interested, and as I open it up to the audience, I'm actually going to do a second polling question. So it's just a question about which technology you're using, whether you're using the lamp itself, whether you're still using qPCR, next-gen sequencing, others, or at the moment, none at all. So I just leave that open for a few seconds, and I'll actually start with the first question. The first question that's come in. I know you mentioned it, but perhaps you could just expand very, very slightly. And somebody's asking, can I use a multiplexing assay?
Yes, so in LAMP, yeah, you can use the multiplexing, Steve. So as I mentioned before, so usually on LAMP, you can use like 2 or 3 targets, because increasing the number of primers, you can have a non-specific amplification. Right, right, excellent. Yeah, just share the poll with you as it's come through. So a lot of you, very nicely, are using the LAMP technology. So 56% are using the LAMP technology, 29% are using qPCR at the moment, and then 2% another, and 13% nothing at all. So that is interesting. So now what I'm going to do, I'm going to bring on the other questions. So first of all, there is a question that's come in, and it's regarding the difference between air drying and lyophilization. So actually, I'll fill this question because it's a question I often get asked. And I think what the person is asking is, can you use exactly the same mix for the air drying and for the lyophilization? So can they be used in exactly the same way? And I can tell you the answer to that is no. So the whole idea of lyophilization, first of all, is to take your solution and freeze it. You freeze it, and then you use a vacuum to draw the moisture off. So you need the excipients in there that are going to protect it against the freezing. So we have those in there. For the air drying, we simply take the solution, put it in a convection oven, and heat it up. And so you actually need the complete opposite. You need those excipients that are actually going to protect against heat. So they are different. So the mixtures themselves are different. You need either one or the other. But very good question. Second question is talking about the drying. How long are these mixes dryable for? So obviously, how long can they be stored? So yeah, what I was showing before, like they can be stored at room temperature at least 24 months. So it's so both the two methods, the air dryable and the lyophilized, right, master mixes. Right. And when you say room temperature, you mean ambient temperature? Ambient, yeah. You room like temperature, so I know you can temperature. Depending on, yeah. Next question is, sorry, I asked that one. It's how can the mixes be optimized? So obviously we've got the mix, we want to try and make it better. How do you actually do that optimization? Yeah, so you showed on the slide before, so like we optimize the mixes, like they come like they're really fast, like with the TTRs compared to the competitors. Right. So the master mixes are already optimized. So what you can optimize is the concentration of mag, of magnesium. Right. So that depends on the assay of different assays. So yeah, so the master mixes are already optimized, and depends on the primer you need an optimization of the magnesium concentration. Right, we've actually got some more questions coming in, and they're very, very small. I apologise for this. Not sure how I can draw this out. Bear with me just a second. If I do, so somebody's asking, how is the amplicons detected? And this goes back to the detection methods. So, so the detection method is with intercalated dye. Right, right, and that's the one you recommend. Yeah, yeah. Is there any particular dyes that may cause a problem with the system? Yes. So there are some intercalated dye. It can bring an inhibition, so the reaction. So yes. Right, right. And another next question, I guess this goes back to the optimization as well. How do you can control
false positives with lamp?
So you need to lamp, because has huge amplification and the yield of lamp is really huge. So I recommend it to use everything really sterile and to have small aliquots of everything.
Right.
To have only one use at a time in the reaction.
Right. Right. Next question: have you used other inhibitors, such as inhibitors for blood?
Yes, we are developing master mixes for blood.
Right, and I will actually say
different specimens.
We're going to have a blood lamp mix out in a few weeks' time, and if anybody is interested in testing it, please get in contact with us, and you can see my email address at the bottom. Let us know, and we'd love to have people test it, because we are very confident, and we've worked with a lot of these mixes, and we're very confident it works, and we want to get it into your hands as quickly as possible. Next question is, bear with me, these are very small. It's asking about the air drying. So basically, do you offer help with assay development with regards to optimizing the lamp primers, concentration, temperature, etc., when using dried or lyophilized lamp products?
So yeah, so you need an optimization of the set of the primer. So it doesn't matter if it's air dry or lyophilized or wet. So right. You can do an optimization. So you can use the same primer for both the method, the air dry or lyophilized.
Yeah,
mixes.
Again, I would say I just interject here and just say to the people that are listening, if you are optimizing and you are having problems, you're very welcome to contact us. You will get in contact not just with myself, but with Elder and the other people that actually work directly in the lab, and we will actually be able to help you. So feel free to join to ask us any time.
Happy to help.
Always happy to help. Next question here: do you recommend using DUTP?
So yeah, there are master mixes which they use. Our master mixes, they don't have DUTPs. So we optimize the master mixes. They are all without magnesium, which it helps them to don't have a contamination later.
Right. Yes, that makes sense. And we have a question here. How was the sensitivity of the air dry versus lamp? So is there any difference? So obviously we've showed liquid against lyophilized and liquid against the air dried. So what's the difference between the air dried and the lyophilized?
There is no difference in both the two methods.
Right. So it's up to the customer whether they want to air dry or they want to use lyophilization. Yeah. And certainly they both, I know, have advantages. So the advantage, obviously, with air drying is it's less expensive. It's very easy to do. But of course, with lyophilization, and
the timing is different compared to air drying. It's like faster.
Yes. Yeah.
And the lyophilization, it takes more time to.
Yes. And actually, that comes to another question, and that's the actual mixes themselves when they're dried down. What's the difference between them? Obviously, I think what they're, again, I'm interpreting what somebody's actually asking here. You obviously have a cake for the lyophilization.
Yeah.
What do you have for the air drying, and how different are they?
So they are different. In the air dry, it's a bit more difficult to see it, but you can air dry with intercalated dye. So you will see the cake, the pellet, in the bottom of the.
Right. And for the actual drying as well, I know that there's different amounts of drying, isn't there, for the two of them? So the lyophilized is completely dry, whereas the air dry is not completely dry. It's still got some moisture in there. So you do have to be careful, I know, with it. When you're actually doing the air drying, you need to do some level of optimization to make sure you get the right drying conditions.
Yes.
Because if you dry it down too much, it's difficult to resuspend. Yes. Yeah. What is the speed of lamp enzymes stroke nucleotides? So basically, how fast is it? They're asking.
And so we have, so depending on the assay and depending on the primers, the assay itself, because depending if you use 4 primers or 6 primers, but on the template itself. So
right, right. But it's, I assume it's going to be similar to a normal polymerase. So it's going to be normal polymerases, good polymerases are about 15 seconds per kb, so they're really, really fast. I assume it's going to be a similar type of thing for.
Yes, it's around like 1,000 nucleotides per minute.
Oh, right, right, yeah. So it is fast.
Yeah.
Another question here is, and it seems to have disappeared again, I apologise. It was about the what is there, or is there an advantage of glycerol-free BST over normal BST?
So the advantages of glycerol-free is for the method, the air dry and lyophilization method.
Right.
So for the wet format, you can use both the two, like glycerol-free or with glycerol BST.
Right, right, yeah. Another question here is, what is the temperature of the storage for the liquid lamp master mix?
We store it at -20.
Right, right. So it's like a standard.
It's standard,
yes. Yeah, and that makes sense, I suppose. With an intercalating dye, what's the preferable intercalating dye? So is it cyber green? Is it?
I prefer or more than the Cyto 82.
Oh, Cyto 82. Yeah. Right. Right. Okay.
But yeah, you can use different intercalated dye.
Right. Yes. Somebody's mentioned about warm start. Obviously, NEB have warm start. Do we have anything similar to warm start?
So for the master mixes, so they are all optimized without the cofactors. So in this case, without magnesium. Right. So you don't need a warm start BST.
Right. Right. So
everything starts when you rehydrate, and when you add so we add the magnesium in the end of the reaction when you resuspend the cake.
Right. Last question I can see here is regarding working with RNA and working with DNA.
Yeah.
Can they just use the RNA kit for everything?
Yes. Yeah. The RNA kit, you can use it for DNA or RNA detection.
Right. Right. Okay. So I think that's probably a very, very good place to stop. And first of all, I'd like to thank Elda
thank you, Steve.
for giving us that wonderful information. I'd like to thank the audience for your wonderful questions, really good questions. You will be able to get a copy of this. We'll send out a copy of this presentation to you. Again, I'll apologise for the beginning of it where the slides stopped moving. I'm glad that Elda noticed and she flagged it up for me. So thank you very much, Elda. And it just leaves me to leave you with the last polling question. And it will just be about how you thought about this webinar. Was it useful? Was it unuseful? Neutral? So if you have any questions, you've got my address at the bottom there. Please let me know. If you do want to try any samples, so please get in contact with us. And if you run into any problems, if you want any help with optimization, anything like that, please contact us and you will get through to a proper person, not somebody like me that reads from a script, but to a laboratory person that actually day-to-day works with these. So thank you very much and have a good day.
Thank you, everyone.
Thank you.
Thank you, Steve.
Bye.